Thanks for coming to this webinar today. And we have Aurora Gómez Durán. She's the coordinator of the group Mitochondria, comunicación celular y estrés oxidativo from the SEBBM. And well, she's going to introduce us the two speakers that we have. Thank you, Aurora. Whenever you want.
Hello, good morning. So today we have two speakers that will be talking about regulation of mitochondrial DNA, basically. The first one is Luis-Carlos Tábara. Luis-Carlos did his PhD at Ricardo Escalante Lab in the Instituto of Investigaciones Biomédicas Alberto Sols, where he focused on autophagy and interorganelle communication. And then in 2018, he moved to the mitochondrial biology unit in Cambridge, where he joined Julian's Prudent lab. Julian's lab is focused on mitochondrial dynamics, and his postdoc was mainly focusing on mitochondrial deregulation. Now he's coming back to Spain, but I'm not going to do the spoiler where he's coming back, but mainly he will study how mitochondria communicate with other organelles in different models. So please, Luis Carlos, thank you and welcome.
Okay. Thank you very much. Can you share see my screen now?
Yes, yes.
Okay. Okay, thank you very much for the introduction, Aurora. It's really a pleasure to be here and share with session with you and with David. And as the title suggests, I will be talking today about the interplay between mitochondrial dynamics and mtDNA recycling. So most likely, mitochondria is the powerhouse of the cell because of their central role in energy production through the oxidative phosphorylation process. However, we need to be aware that mitochondria is much more than that. They are involved in many other functions, for example, calcium homeostasis, cell survival, cell death, immunity, senescence, cancer, and even stem cell reprogramming. That's why this organelle is considered a signaling organelle that virtually impacts every cellular function. Because of this, it's very important to consider a global view of the mitochondria, and it has been very nicely represented in this review. And what the authors want to transmit with this cartoon, and I quite agree with them, is that until very recently, mitochondria have been seen, let's say, from a fragmented perspective. So what I mean with that is that depending on our technical expertise, on our scientific background, we used to focus on one particular function of the mitochondria and forgot about the others. However, I think the field is moving forward to have up to a more integrative perspective of the organelle, and this is really good. So to have a very integrative perspective of the organelle is very important to consider the cellular context of the mitochondria. And the cell is full of other organelles. This is a beautiful video from the lab of Jennifer Lippi-Consuar in Janilia, where they use this cool technique called multispectral mixing to visualize multiple organelles in one single cell. So what we can see here in this video, where the mitochondria are in green, is that these organelles are constantly interacting with the other organelles in the cell. And nowadays we know that these interactions are essential not only for mitochondrial function, but also for cellular physiology in general. And I think in the session of today, both David and I'm going to focus on one particular interaction between mitochondria and degradative organelles such as lysosomes or autophagocytes. But before going there, we need first to introduce properly the central organelle of this talk, the mitochondria. Mitochondria are very complex organelles. They have two membranes, the outer and the inner mitochondrial membrane, separated by the intermembrane space. On the outer mitochondrial membrane, we can find, for example, the SAM and the TON complexes that are the main entry gates for nuclear-encoded proteins. Actually, we need to remember that 98% of the mitochondrial proteome is encoded in the nucleus. This demonstrates the huge investment that the cell does to maintain mitochondrial physiology. Here we also have the mucous complex. So these mucous physically breach both the outer and the inner mitochondrial membrane. And inside we have the mitochondrial matrix. Here is where the inner mitochondrial membrane invaginates, forming these amazing structures called cristae. You can also nicely visualize the cristae here in this electron micrograph on the right. So here in the cristae is where the respiratory chain and the ATP synthase are located, and therefore is where ATP production happens. I would like to share with you also this fantastic paper published a couple of months ago in Science. This is the first time we're able to visualize all this complex in native cells. It has been done in chlamydomonads. So here, for example, we can see the ATP synthase in blue accumulating at the tip of the crystal, where the respirasomes are more concentrated in this flattened area. We can also see other complexes inside the mitochondria, for example, HSP60, that is a chaperone, the mitoribosome, also probitin that act as kind of a scaffold and is very important for IMM architecture. So this paper is really fantastic. I would really recommend to read it. It can be a breakthrough in the field, actually. But we are still missing here a key component of mitochondrial physiology, that is the mtDNA. So here I put some of the main characteristics of the mitochondrial DNA. That actually is probably the most iconic attribute of this organelle, that they have their own genome. So it's maternally inherited, it's located in the matrix, but very closely associated to the inner mitochondrial membrane. This will be important for the talk of today. And in humans, it's very small. It only has 16.6 kb and encodes for 37 genes. And among these genes, we can include 13 OXPHOS components that are essential for ATP production. And actually, when any of these OXPHOS components encoded by the mitochondrial DNA are malfunctioning, we have a total collapse of the OXPHOS process and we have energy depletion. Something really important also is that mtDNA replicates independently of nuclear DNA, independently of the cell cycle. This is a process that we know as relaxed replication and leads to high copy number per cell that can range between 100 to 1,000 copies, depending on different tissues and energy demands. So for example, here in this picture, we can see the mitochondria in green, and each of these foci in magenta represent one nucleoid, and one nucleoid has an average of 1.4 mitochondrial DNA molecules. So yes, the important thing here is to have in mind that the mitochondrial DNA is a multi-copy genome. Because of that, because it's constantly being replicated, mitochondrial DNA is very prone to have mutations. It has a high mutant rate, and this leads to a condition that we know as heteroplasmy. And heteroplasmy reflects the existence of both wild-type and mutant mitochondrial DNA molecules in the same cell. Importantly, some of these mutations are pathogenic, and when the amount or the percentage of these mutations surpass certain threshold that in the field we know as biochemical threshold, mitochondrial dysfunction will happen, leading to disease. So depending on the region of the genome that is mutated, the patients can experience different symptoms. I'm not in a good position to say that one symptom is more important than the other. All of them are very significant, but probably the most common ones are neurodegeneration, muscle weakness, and some types of optic atrophies. If we talk about mitochondrial DNA and disease, we should not forget mitochondrial DNA depletion syndromes that are marked or are characterized by a marked reduction in the levels of mitochondrial DNA. In this case, they are not caused by mutations in the mitochondrial genome, but by mutations in nuclear genes that somehow affect mitochondrial DNA maintenance. And actually, most of these genes are involved somehow in mitochondrial DNA replication. So in any case, both heteroplasmic diseases and these depletion syndromes are terrible conditions with very poor prognosis, and that's why it's really important to understand the dynamics of the mitochondrial genome in order to find new treatments and cure for these patients. So if we want to understand the dynamics of the mitochondrial genome, we need first to understand the dynamics of the organelle, the dynamics of the mitochondria. This is a super resolution live cell imaging from the lab. We used on 20 tag with the state goal. A state goal is a brighter version of a GFP. It's really good if you want to do long videos because it has a great photostability. So here we can observe that mitochondria are highly dynamic organelles. They are constantly moving around throughout the cell. They fuse, they divide. This is a really fascinating dance. And what is more important is that this transition in shape are essential for cellular homeostasis. For example, mitochondrial hyperfuse their network under mild metabolic stress. This happen to increase oxygen, to increase respiration, to resist adverse situations that can happen to the cell. On the other hand, mitochondrial shift to a more fragmented scenario under more severe metabolic stress. We can imagine, for example, in this context, hypoxia, also during apoptosis, but also it happens during more physiological conditions. For example, mitochondrial quality control. We will see this later in the talk of today. Also during mitochondrial motility, For example, this is very obvious if you think about neurons. You want the mitochondria to be smaller to facilitate the transport of the organelle through the long axons. And of course, during cell division, you need to divide to fragment the mitochondria to ensure an equal distribution of this organelle to every daughter cell. However, what is relevant for the talk of today is that mitochondrial dynamics are also essential for mtDNA homeostasis. In fact, it's very well described in literature that defects in either mitochondrial fusion or fission leads to the loss of mitochondrial DNA. On the other hand, maintaining a basal degradation of mitochondrial DNA through mitophagy is also very important to prevent an abnormal accumulation of molecules. If this happens, we can have different stress situations. For example, the accumulation of deleterious mutations that will be detrimental at the end for OXPHOS function and ATP generation. It can also happen that this leads to a situation of mitonuclear protein imbalance. That means an imbalanced expression of nuclear and mtDNA encoded proteins, which is pretty bad for cellular proteostasis. And eventually, if we have too much mitochondrial DNA molecules inside mitochondria, it can also happen that they will leak into the cytosol, where they can activate the SIGA-testin pathway driving inflammation. So despite the critical importance of this process of the autophagic degradation of mitochondrial DNA, the players involved are very poorly characterized, and this has been the main focus of my research in the last years. So if we want to talk first about the process of mitochondrial degradation in general, is what we know is that the process starts at the periphery of the organelle, where damaged regions segregate into smaller mitochondria that are targeted through lysosomal degradation by the mitophagy process. So most people investigating this process of mitochondrial quality control do that in different contexts of biochemical dysfunction, such as the loss of membrane potential, oxidative stress, or hypoxia. However, how internal mitochondrial membranes, such as damaged cristae, are sent for degradation remains a very outstanding question. And understand this question will be essential to understand the question of today about how mitochondrial DNA is being degraded, because we need to remember that these molecules are both structurally but also functionally associated to this mitochondrial cristae, and we will see this later. So with this in mind, I'm very happy to present today our recent discovery, where we identified this protein called NTFP1 as a critical regulator of the quality control of the inner mitochondrial membrane and the recycling of mitochondrial DNA molecules. So who is this guy? So NTFP1 is an inner mitochondrial membrane protein that has been associated to the regulation of mitochondrial morphology for nearly 20 years. In this way, the absence of NTFP1 leads to mitochondrial hyperfusion, whereas NTFP1 overexpression triggers a dramatic mitochondrial fragmentation. However, how does NTFP1 regulate mitochondrial dynamics was unknown, and this was the first question I wanted to tackle when I arrived in Cambridge some years ago. So the first thing we did is analyze how exactly the absence of NTFP1 affects mitochondrial morphology. So today I'm using U2S as cellular model. I will be showing also a lot of image from mitochondria. For this, I'm using atom-20 antibodies. So this is endogenous marker. And also, importantly, as a positive control of mitochondrial morphology, I'm using the inhibition of GRP1 that, for those ones that are not familiar with mitochondrial dynamics, is the major regulator, is the master regulator of mitochondrial division. So here in this picture, we can see on the left, in a control situation, we have a mix of a small and more or less intermediate mitochondria. As expected, when we inhibit GRP1, we shift this to a very elongated mitochondrial phenotype that is characterized by this increase in mitochondrial length that we quantify here in this graph on the left. However, when we inhibit NTFP1, we have something completely different. Instead of this massive increase in length, we have an increased number of mitochondrial interconnections that I highlight here also in red. So this increase in mitochondrial interconnections that I quantify here in this other graph, you can see clearly the difference between GRP1 and NTFP1. This increase in connections is what we know as mitochondrial hyperbranching phenotype. So I think it's very obvious if we compare these two images that these two proteins regulate mitochondrial morphology through different pathways. So based on this difference, we ask if actually NTFP1 was involved in mitochondrial division. To do so, we use super resolution light cell microscopy. With this kind of technique, we are able to discern midstream fission events that are those ones happening in the middle of the organelle, as you can observe here. And they are mainly involved in mitochondrial biogenesis and peripheral fission. In this case, is this kind of asymmetric fission event that results in the formation of this small and round-shaped mitochondria, and this process is mainly involved in mitochondrial degradation. Here we will see now another example. Yeah, here we go. So what we did is quantify these two events in the presence and absence of NTFP1, and our quantification revealed that midterm fission rate was unaffected in NTFP1 knockout cells. However, we have a significant reduction in the number of peripheral fission events. So this data suggests that this protein NTFP1 is not strictly required to drive division because it can still happen when this protein is not there, but it may play a role in the segregation of these small mitochondria that are targeted for degradation. So we need to remember that mitochondrial dynamics is always a balance between fusion and fusion. We have seen fusion. Let's see now what happened with the fusion process. In this case, we can distinguish between a stable fusion that happens when the two mitochondria remain fused over time. We use a threshold of 1 minute or more than 1 minute to consider a fusion as a stable fusion event, and transient fusion. Here, if you focus in this yellow area, you will see that the fusion event is rapidly followed by a division event in the same area. So in this case, what we did is quantify the duration of the fusion events in wild-type and NTFP1 knockout cells, and what we found is that fusion events last longer when NTFP1 is not there. This translates into an increased rate of stable fusion events and a decreased rate of transient fusion. So at this point is when we start thinking that probably this hyper-branch phenotype I talked a couple of slides ago was due to an increase in mitochondrial fusion. So to understand better how this protein can control the fusion process, we decided to look at OPA1. So OPA1, remember also that we are talking about NTFP1 that is an inner mitochondrial membrane protein. So in the process of inner mitochondrial membrane fusion, we have two main regulators that are OPA1 and cardiolipin. So the current model suggests that this protein, OPA1, is recruited to cardiolipin-enriched microdomains where it oligomerizes. And this oligomerization drives the tubulation of the two opposing membranes that will come close together and finally will fuse. So we decided to study OPA1 oligomerization regarding NTFP1 expression. So we found using BMH, the cross-linker BMH, we found that, for example, here on the left, that two different knockouts for NTFP1 have an increased amount of OPA1 oligomers, suggesting that when NTFP1 is not there, we have an increased fusion capacity. However, when we overexpress the protein, when we overexpress NTFP1, we have the opposite phenotype. You can observe here in this plot on the right that NTFP1 overexpression leads to a decrease in OPA1 oligomers. So this data suggests that somehow NTFP1 prevents mitochondrial fusion by dampening or inhibiting OPA1 oligomerization. So this was pretty interesting. So we then asked in which context NTFP1 is playing with mitochondrial fusion. So to understand this, we decided to follow NTFP1 dynamics using live cell imaging. So in the talk of today, I will be showing you always NTFP1 in magenta. Here we can observe that this protein tends to accumulate at the periphery of the organelle, leading to the formation of these enriched microdomains. If we pay attention to this video, you will see that these peripheral regions eventually segregate from the network, leading to the formation of these small mitochondria enriched in NTFP1 that we know also by the acronym of SMEL, small NTFP1 enriched mitochondria. So please remember this acronym SMEM. I will be using it quite a lot in the talk. So what it was particularly fascinating about this SMEM is that once they leave the network, they are not able to fuse back, never again, suggesting that somehow the presence of NTFP1 or at least high levels of this protein inhibits mitochondrial fusion. So what's going on with this mitochondria? Since they are physically isolated from the network, our first idea was, okay, they should be targeted for degradation. And actually that was the case. And in the next slide, I'm gonna summarize 1 year of work where we delineate the autophagic pathway of this small mitochondria enriched in NTFP1. So we found that first, after segregation, these SMEM are targeted by the ubiquitin proteasome system that drives the degradation of automitochondrial membrane proteins. That's why you will see that many of these SMEM, of these small mito, are negative for the OMM marker TON20. This facilitates the exposure of the inner mitochondrial membrane to the cytosol, and this acts as a signal to recruit the autophagic receptor P62, which later on will recruit the autophagic machinery. Here, for example, you can see how nicely these LC3 positive vesicles decorate the SMEM. And once the autophagosome is completely formed, it will fuse with the lysosome that we stain here with a LAMP1 antibody for degradation. So I think in this slide is pretty well summarized. It's very clear that these small mitochondria are target for autophagy, and specifically they are target for autophagy in a ubiquitin p62 dependent manner. So this was very cool. So the next question goes, of course, which kind of cargoes are transported inside for degradation? Which kind of cargoes are sent to the lysosome? Is any kind of cargo selectivity? So in this regard, we found that these SMEN can contain a wide variety of internal mitochondrial membrane components, but what was very interesting for us is that they can contain mitochondrial DNA. For example, here we can see this pre-autophagic mitochondria negative for TON20, enriched in NTFP1, containing a DNA molecule or containing the DNA-related protein TFAM. Of course, we confirm that this small mitochondria containing mtDNA, a target for the lysosome for undergoing degradation, as you can observe here in this other picture on the right. So at this point, we were quite excited because this could represent a novel pathway for autophagic degradation of mitochondrial DNA. So we thought that if this hypothesis was true, what we would expect if we inhibit this pathway, if we block the capacity of mtDNA for go to degradation, we should expect an accumulation of mitochondrial DNA molecules that cannot be degraded. To explore this, we use different methods. Here we use, for example, the double PCR in single cells. So this is a absolute quantification of mitochondrial DNA copy number. And here we observe that the inhibition of NTFP1 for 3 or 6 days leads to a massive increase in mitochondrial DNA levels. It was almost double, or actually double at 6 days, which is pretty extraordinary. We were able also to recapitulate this in 2 different knockout clones for NTFP1. You can observe that these clones present around a 70% increase in mitochondrial DNA copy number with respect to control cells. And this is just another graph showing you that when we reintroduce NTFP1 tagged with flag into the knockout cell line, we are able to rescue this aberrant accumulation of mitochondrial DNA. We also use, because we are microscopists, mainly, we also use a structural illumination microscopy, super resolution microscopy, and a DNA antibody. So with the DNA, we are able to see mitochondrial DNA. So basically, if you are not familiar with DNA staining, each of these foci correspond to 1 nucleoid, and 1 nucleoid on average has 1.4 mitochondrial DNA molecules. So in this image, we can clearly observe, and also in this quantification, that NTFP1 knockout cells present an abnormal accumulation of nucleoids and a normal accumulation of mitochondrial DNA. So altogether, this data demonstrate that our original idea was true, and the pathway we are describing today is important or is required for the basal degradation of mitochondrial DNA. So then we wanted to go a bit further, and we asked if this pathway could also operate not only basal conditions, but also during mitochondrial DNA damage. So to this end, we use the DNA intercalating agent, ethidium bromide, that has been shown actually by David, that we took after me to induce mitochondrial DNA degradation in autophagic dependent manner. So here we have a control cell. So we see that EV treatment for 5 days leads to a drastic reduction in the number of mitochondrial foci. So in the, you remember that each of these foci is 1 mitochondrial genome. So what we observe is that NTFP1 knockout cells were quite resistant to this effect, suggesting that, yes, the pathway we are analyzing is not only important for the degradation of mitochondrial DNA in basal condition, but also for the disposal of damaged mitochondrial DNA molecules. So in the final part of my presentation, I want to talk a bit more about the mechanistic aspect of the degradation of damaged mitochondrial DNA. And to investigate this process, we decided to follow NTFP1 dynamics during mitochondrial DNA damage. So here on the left, we can see that NTFP1 in magenta again is present in control situation a more or less uniform pattern throughout the mitochondrial network. However, upon mitochondrial DNA damage with ethidium bromide, actually this is, I think, is 3 days, we see a strong relocalization of NTFP1 towards the periphery of the mitochondria and to these small mitochondria. So if we decide to go even have a closer look to this using super resolution microscopy, and when we do that, we have something very, very interesting, and is that we found that this NTFP1 tends to form ring-like structures around damaged mitochondrial DNA. Here you see the DNA molecule in yellow, how NTFP1 is kind of surrounding them. So for us, this was a very reminiscent of the concept of the sphere of influence of mitochondrial DNA. So this model suggests that each mitochondrial DNA molecule has a sphere of influence where the DNA-encoded products will be located. Since these products that are OXOS components are essential for crystal integrity, this model predicts that the cell can be able to detect the presence of damaged mitochondrial DNA by detecting alterations in the surrounding membranes. So based on the localization of NTFP1 in these surrounding membranes, we asked, why not if NTFP1 is detecting crystal alterations rather than the mitochondrial DNA molecule itself? And actually that would make sense because our stimuli of mitochondrial DNA damage, acetylbromide, leads to a massive reorganization of the inner mitochondrial membrane compartment. You can observe here that these red arrows, when we treat the cells with acetylbromide, we have the formation of these onion-like crystals that are totally detached from the inner mitochondrial membrane. So to explore this possibility, we decide to alter IMM morphology through other alternative approaches. So we decide to use the inhibition of ATP5 and MYC60. ATP5, of course, is the key component of the ATP synthase, and MYC60 is the major component of the mucus complex, and it's very well known that the presence of either of these two proteins is essential to preserve crystal integrity, as you can observe in these two electron micrographs. So what it was very interesting to observe is that when we damage the inner mitochondrial membrane through the inhibition of ATP5 or MYC60, the number of these small mitochondria being targeted to the lysosome massively increase. So in a control cell, we found 1 or 2 of these men per cell. However, when we trigger crystal damage, the number of those is much higher. You can observe this here, but also in this other quantification. So this experiment was conceptually very important because it suggests that the pathway we are describing today is not so specific for mitochondrial DNA, but it forms part of a more general mechanism for the quality control of the inner mitochondrial membrane, for the quality control of the crystal. So to corroborate the role of NTFP1 in this pathway, we decided to use the mitophagy reporter cell line mitochema. So to put it simply, with this cell line, we're able to visualize the mitochondrial network in 2 different colors. So when mitochondria, they are healthy, they're in the cytosol, they will be in one color, in this case, cyan. And when mitochondria are undergoing degradation in the lysosome, they will be another color, in this case, are represented in magenta. So in this image, we can observe that when we trigger crystal damage through the inhibition of ATP5 and MYC60, we have a strong mitophagy response in wild-type cells, as evidenced by the increased number here of these magenta dots, an increased number of mitolysosomes. But this is not happening in NTP1 knockout cells. Here is also the quantification of this image. So this data clearly demonstrate that the presence of NTFP1 is not only essential for the degradation of mitochondrial DNA, but also for the autophagic targeting of defective crystal. So this was everything for me talking about results. So this is the final model. We thought that, or we think that NTFP1 serves to segregate damaged crystal into small mitochondria. This is small mitochondria because they are highly enriched in NTFP1. They have antifusogenic capacities and they become isolated from the network. They cannot fuse again. And they are eventually target for autophagic degradation in a ubiquitin p62 dependent manner. What I think is particularly cool about this model is that it links the quality control of the crystal, the quality control of the IMM, to the recycling of mitochondrial DNA. So if you have questions, you can also have a look to the paper that was published some months ago. And now I just need to say thank you to all people that have been with me in the lab. In the last 7 years, I've been working as a postdoc, as Aurora said, in the lab of Dr. Joran Prudent at the Mitochondrial Biology Unit in Cambridge. I want to thank especially to Roy that helped a lot during the revision process of the paper to acquire some super resolution image, and also to our collaborators in the MBU, to the group of Patrick Chinnery, especially to Stephen and Michele that helped me a lot with the digital PCR for mitochondrial DNA copy number analysis. Also, of course, to the funding agencies. And also, I'm very happy now to say that I will be opening my own lab in October 25. We are moving to this beautiful building, to the CRG in Barcelona. So we will start in October 25. I'm trying to, actually now I'm starting to try to build a team. So if you're interested in a PhD or a postdoc, or would you like to collaborate in some aspect of mitochondrial dynamics, just drop me an email. I will be more than happy to discuss potential collaborations or opportunities. So in the lab, will be focused mainly the first years in study the cell biology of mitochondrial DNA. So we have several questions we want to answer. For example, what are the mechanisms controlling the distribution of mitochondrial DNA, similar to what have been seen today? How does the cell detect the presence of damage of mutant mitochondrial DNA? And also, we will be very interested in understanding the physiological adaptations of the cell to mitochondrial DNA damage. So that was all. So I'm happy to take any questions. Of course, thank you for attention. I hope to see you soon.
Thank you very much, Luisca. Very nice talk. So does anyone in the audience have a question? Okay, we have a question here. Good morning. Thank you for the nice talk. I have several questions. Do you know anything about how MTFP1 is regulated? Do you assess the inflammatory state of the cell upon silencing or overexpressing MTFP1?
Okay, so thank you very much for attending. So regarding the first question, this was a paper that published actually by Julian some years ago in Molecular Cell that said that NTFP1 is regulated by mTOR. We have also there is 2 or 3 papers saying that NTFP1 can be also regulated by hypoxia. This is also quite interesting if we think about the role of NTFP1 in the quality control of the inner mitochondrial membrane, because hypoxia at the end is another kind of mitochondrial damage. So it can be like just that oxygen can regulate somehow NTFP1 expression. And regarding question 2, this is a very interesting question, and this is something that we are actually now evaluating in the lab, not only for NTFP1, but also for other proteins, how mitochondrial DNA imbalance can promote inflammatory phenotypes. This is something that is ongoing work. Yes.
Thank you. Any other questions? So I have a question, since we have a lot of students here, I presume. And students, when they see microscopy images, they think that they are seeing things that are wrong. Okay, so how do you come with the peripheral fission rate? I mean, how many hours did you spend looking at those mitochondria breaking? You know, I think it's important for everyone here in training, so that they have to trust what they see, or it's about observation.
Yeah, I mean, of course, you need to have the eye, like how do you say, like the eye of the lynx. You need to be trained a lot of hours to do this kind of analysis. But regarding the midzone and peripheral fission events, what I would like to point out is that I think you cannot do this very properly with normal confocal or scanning microscopy. You really need to have super resolution microscopy. I mean, you can have an idea with confocal microscopy about peripheral and midzone, but if you really want to distinguish peripheral versus midzone, I think you need to this kind of high resolution microscopy. Otherwise,
no, definitely. But in any case, you will have to you will have to have the eye and spend a lot of hours looking
yes,
at
I
where
don't
this
know.
drop is going, right?
I will say that in the last 7 years, I will say that an average per week, I will spend like, I don't know, 15 hours per week watching mitochondria, at least.
Yeah. Well, I think that's little.
Yeah. I think much more, yes.
Anyway,
Yeah,
so
I have a question. Yeah, when you have mutations in the mitochondrial DNA, do you also have changes in energetic metabolism?
Yes, of course. Yes. Just
How
because
does it affect? Like, I have first energetic metabolism, but then, for example, in cardiolipin levels, does it influence somehow?
you're asking if somehow the mutations in the mitochondrial genome can
Like
influence cardiolipin.
glucose.
Yes, of course. So you have mutations in the mitochondrial genome, depending on the region that is mutated in the mitochondrial genome, the mutation will affect different complex of
Yeah.
the oxidative fluoridation process. So you can have different type of damage.
Yeah.
But yes, the common factor to all these mutations the common factor of mitochondrial diseases at the end is that energy depletion. However, it's true that the field is moving forward to say that it's not only about the depletion of ATP, that many of the phenotypes of the patients
I
are
know.
also due to maladaptations coming from these defects in ATP production. And regarding cardiolipin, yes, of course, there is a clear interaction between cardiolipin and mitochondrial DNA. We know that actually mitochondrial DNA replication, there is some paper suggesting that because mitochondrial DNA is in the matrix, but closely associated to the mitochondrial membrane, there are papers saying that the cardiolipin can serve like an anchor or something like where the mitochondrial DNA will attach to start the replication process. So of course, there is a cross interaction between lipids and mitochondrial DNA replication. Yes, and it's very well known, for example, if you disrupt cardiolipin in Barth syndrome patients, for example, you have the depletion of mitochondrial DNA because mitochondrial DNA cannot replicate properly without a proper lipid composition in the inner mitochondrial membrane.
Okay, that's interesting. Yeah.
Okay, thank you very much, Luiska. It's been wonderful. I have one. Okay, we have the last question, but because we are on time now. So one more question. Sorry, it's a bit more general and less related. Why and how does mitochondrial DNA start to leak upon his accumulation? And what exactly is the effect of its copy number in the mitochondria?
Okay, so regarding the first question, it's something like, of course, it's a hot topic, and I think many people is investigating that. Like the topic of mitochondrial DNA release has been like on fire the last 10 years, I would say. We have several mechanisms. We have back pores, we have limited MAM, we have BDAC pores. Now in the lab, my colleague Vincent Pop also published in collaboration with Christian Frexa that they can pop out through MDBs generation. So depending on the kind of damage the mitochondrial DNA can suffer, I think different mechanisms for release will undergo. So if we are talking about how the accumulation of mitochondrial DNA leads to the leak of these molecules to the cytosol, as I just said before, is something we are working on. And the quick answer for you is that we are evaluating all these pathways, and still we don't have a proper answer. So what does it mean is that I think from my personal point of view is not that you have only one mechanism of mitochondrial DNA release for one specific situation, but several of them can happen at the same time. It's not only that you have backspore and will be backspore dependent and not BDAC. I think it's a mix. So if you have a situation of stress, at the end, the mitochondrial DNA will go out by any mechanism. That's my personal idea. And what is the effect of this on copy number of mitochondria? This is something very interesting. I don't know if I get the question well, but it's like, are you proposing that if you have a leak of mitochondrial DNA, how the mtDNA is maintained at the end because you have a leak of molecules outside? So I think what could happen in this kind of situation, you will need to increase mtDNA replication to be able to restore mitochondrial DNA levels. But this is a really interesting question. I think nobody has actually.
I think David is going to answer some of these questions later on.
Oh, yeah, probably, yes.
No, well, I have my own mechanism, but I totally agree with what Luis-Carlos just said. I think it's actually depending on the type of stress that you are or that we are using, that the mechanism can be one of another one, and then not just one exclusive mechanism.
Yeah.
And I will show you something about in our type of stress, let's say.
Yeah.
And I will spoil. This is mutation DNA mutation specific. That's it's not only damage. Each mutation behaves differently.
It could be as well.
Even in the same complex, it's not that ATP is how the cells reacts to the given mutation. How much of this mutation it has is much more complicated than that. Well, that's why we are here, right? Anyway, so thank you very much, Luiska. And now is the time of David. David did his PhD in Valencia, and then he moved to Germany, to Cologne, where he was first studying mitochondrial RNA granules, and then he moved to Rudi Bisnoff lab, where he started to study mitochondrial DNA. He had a little starting position in Cologne, and now he's in the University of Düsseldorf, where he tried to study mitochondrial DNA maintenance, aging, and he's going to talk about this today. Thank you very much, David, for joining us. It's a pleasure.
Good. So let's see if I do it correctly now. Share.
Perfect.
Perfect. Okay. So thank you very much for this kind invitation and to allow me to show this talk to mostly a Spanish audience, no? Because I'm not really used to to do that, no? But it's a pleasure to be here. I'm so lucky also that Luis-Carlos talked before me, because he introduced a lot of topics that I could skip right now, or maybe not talk so much about that, and more focus in our results, no? So we have heard already, and we know, I think this is a kind of a school biology, that mitochondria has its own DNA, no? So it's a very small molecule, during millions of years of evolution, has been reduced to a very small molecule containing only, in the human mitochondrial DNA, 13 protein coding genes, and then a bunch of RNAs, transfer RNAs, and ribosomic RNAs, which are actually important to translate the proteins encoded in the mitochondrial DNA. Although the mitochondrial DNA is really small as a molecule of DNA, it's also prone to accumulate mutations or to accumulate damage, as it happens for the nuclear DNA. This damage occurs just basically by normal function of the cell. There might be replication errors. There might be oxidative stress just produced by normal function of mitochondria. And there might be other chemical changes. So all these changes might modify the structure of the mitochondrial DNA, interfere with replication, interfere with transcription, and then just produce mutations, no? One of the things that we know, and Luis-Carlos said before, is that mitochondrial DNA has many copies. So there is not only 2 copies like the nuclear genome, we have hundreds to thousands of copies depending on the cell. This means that the cell more or less tolerates the damage. So it doesn't matter if you have some 20%, or maybe not 20, but a group of molecules which are damaged, as soon as you have some wild-type molecules that can do the work, no? But one very interesting thing, which is known by many years, is that we, humans and all long-living organisms, we accumulate mutations just by aging. And it looks, at least, for example, in this picture I'm showing you, in a skeletal muscle, it looks like there is a barrier at 60 years old when the number of mutations really accumulate exponentially. It is not only the muscle, so there is also in other many tissues, and this is known by many years. For example, you see here, it's known that in retina, in several areas of the brain, so for example, the substantia nigra, really linked, for example, to Parkinson's development, skeletal muscle, as I said, heart as well, sperm, ovary, and skin. And this led to the scientists to develop what is called the mitochondrial theory of aging. And this basically is that damage in the mitochondrial DNA that accumulates during aging affects the tissues in a way that it leads to aging, no? So all the effect that we have during aging, as we age, is caused by, not all of them, but of course a huge proportion, because of damage of the mitochondrial DNA and mitochondrial dysfunction. So starting from this, as Aurora said, I came from Cologne, which is a hotspot in aging research. And during my postdoc, I joined the lab of Rudi Wisner. And I think I was so lucky just to continue a project that started many years ago from another postdoc who was in the lab much before me, who was Olivier Baris, that today is also a group leader in France, as his own group in France. And they generated a mouse model to study aging related to mitochondrial DNA damage. What did they do? So mice also age, no? Mice, they can last like 2 years or something like this, and they also acquire mitochondrial DNA mutations, but the proportion of this mutation is very low. So how can we increase that? So these 2 guys, they arrived to a conclusion. So what we can do is we can just mess up with mitochondrial DNA replication just to increase the number of the mitochondrial DNA damage, or the number of mitochondrial DNA mutations. And they use Twinkle. Twinkle is the mitochondrial DNA helicase, and mutations in Twinkle that leads to diseases, but the most known is progressive external ophthalmoplegia, and also mitochondrial DNA depletion and deletion syndrome. So basically is that there is a point mutation in Twinkle that interferes with mitochondrial DNA replication, and then the replication is not performing properly, and it's causing just the formation of some small molecules containing big deletions in the mitochondrial DNA. It also happens point mutations, no? So it depends a little bit on the tissue that we are talking, but it basically increases the chance of mutation. So we have used this mouse model many years. People before me used it. I use it as well, and we developed many mouse models. And I'm also using this mouse model in my lab right now. The mice are growing, let's say, but we use it in many models. So, for example, epidermis, cartilage, skeletal muscle, heart, extraocular muscle, and also dopaminergic neurons. In all of them, this mutation of twinkle causes like an accelerated aging. These animals, tissue-specific, this tissue is like an aged tissue. Now I'm going to show you something in the muscle, because this is when all the stories start. So here I'm showing you a long-range PCR, showing these deletions of the mitochondrial DNA, which, as you can see, they are strongly accumulated in 24 months. These are very old mice, not so 2 years. They are almost reaching the ending of their life. And we have a lot of deletions in the mitochondrial DNA. So the accumulation of these deletions at the end, if there are too many, they surpass the specific threshold, depending on the type of cells, it develops to mitochondrial dysfunction. And this is actually what we saw by using a COX-SDH staining. So this is a enzymatic staining that can tell us when we have mitochondrial dysfunction in a cell. So here you can see skeletal muscle, different skeletal muscles, soleus, EDL, gastrocnemius, and tibialis anterior. And these are 2-year-old mice, and you can see in some of these they have these blue cells. These blue cells mean they are COX-deficient cells, therefore they contain mitochondrial dysfunction. And the number of fibers containing mitochondrial dysfunction increases with age, which is expected. But surprisingly, this is something that really puzzles us a little bit. It was different depending on which muscle. So it was highly accumulated in for example, here in gastrocnemius, but was also accumulated in tibialis, and was very little in EDL, and also very little in soleus. And this was when I arrived to the lab, and then I said, okay, what can I do with this? So I came from another field, more mitochondrial quality control, and then just coming back, okay, is it possible that mitochondrial quality control defines or controls the accumulation of mitochondrial DNA? And this is not new, what I'm just telling you. So mitophagy was discovered in 2004. It was originally discovered in yeast. In 2008, it was discovered that it also exists in mammals. And already in 2005, even before it was described in mammals, it was already proposed that mitophagy might play a role in retarding the accumulation of somatic mutations of mitochondrial DNA with aging. So it was always believe that the removal of mitochondria might control the accumulation of mutations. So starting with that, we invest a lot of money, a lot of time, a lot of effort to develop or to grow in our lab and to generate a mouse called mitotimer. So mitotimer is just a fluorescent reporter that can change from green to red depending on the oxidation status of the mitochondria. And also red mitochondria means that this mitochondria is oxidized and it's undergoing mitophagy, similar to the schema reported that before Luis-Carlos was showing. By our surprise, and after measuring many muscles and many time points, we did not see mitophagy. So these are 24 months old in the soleus, for example, but we also have that in tibialis. And we, of course, we see mitophagosomes, but we cannot see any difference of mitophagy. So that was a little bit disappointing, because as I said, we invested time and effort and money and everything, but what the hell was going on there? So in parallel to this experiment, we were also working in cells, and we generated cells expressing this twinkle mutation. And here as well, we use this reporter that, again, is similar to, it's not the mitochema, but it's similar to mitochema. It also shifts colors depending on the mitochondria. If it's in the normal mitochondria is green. If it goes to mitophagy in the lysosomes, then it turns to red. In that case, I label it with cherry. We don't see any mitophagy activation when we interfere with mitochondrial replication by expressing twinkle. We do see mitophagy when we messed up mitochondria with antimycin oligomycin. So it was kind of similar to the effect that we see in mice, but still, I'm very stubborn, and I never give up really quick. And there was one thing that made me believe that something was going on. The thing is that when we express this twinkle mutation in cells, we get mitochondrial DNA depletion. And this is very well known. When we are using it in replicative cells that they replicate quite fast, we don't get deletion, we get depletion. However, when we treat these cells with different inhibitors of quality control, in that case, autophagy. So we use 3 different inhibitors inhibiting different steps of the autophagic process. We use 3MA, this guy here that has this horrible name I'm not going to say, and chloroquine that blocks the lysosomal degradation. So in all cases, when we block the lysosomes, we could recover mitochondrial DNA copy number. We could recover this depletion that we are getting, we recover it. So this, in my opinion, this means that the lysosomes were actually involved in doing something here, no? And they are involved in this depletion of the mitochondrial DNA. So it might be that it was just fastly removed. So, but how can we know what's going on there? So this picture has been shown before, because also I use it a lot in my talks about the sphere of influence. This was actually a very important paper from Christoph Osman in München, and he described actually the sphere of influence. So one nucleoid can actually control the biochemical properties of the area where it is located. So the question here was, can we know what is this area of influence when we have mutated nucleoids and compare it to a wild-type nucleoid? Does it change? So at this time, also it was developed a technique that now we use quite often. It's called proximity biotinylation or proximity proteomics or spatial proteomics. There are many variants, and I'm going to tell you in this talk 2 of them, but this I'm going to start with APEX2. APEX2 is a molecule that you can just fuse by normal, traditional genetic engineering techniques to any protein that you want. In that case, we fuse it to twinkle. We fuse it to twinkle, to wild-type twinkle, and to the twinkle carrying this mutation. APEX2 is able to, by a chemical reaction, to biotinylate all the proteins in the surrounding area. When we biotinylate these proteins, we are able to isolate them, and we can analyze them by mass spectrometry and therefore compare what's going on in the area of influence of one nucleoid with or without mutations. No? But another thing that is very interesting for APEX2 is that we can also use it in electron microscope. We can actually depict where are the nucleoids located. And thus, let's start by this. No? So here you can see that what we did is so we take twinkle, APEX2, and this is a normal, these are C2C12 mouse myoblast. This is a normal cell without APEX2, beautiful mitochondria. This is a positive control expressing a matrix targeted APEX2. You can see a dark precipitate exactly in the matrix that is not dark in the crystal. And you can see here twinkle with APEX2, where you can see that this black precipitate really concentrate in some areas of the mitochondria, which we know that nucleoids are there located. What happened then? So, okay, we did that experiment, of course, with wild-type twinkle, mutant twinkle, and we saw these weird structures. And this is actually very interesting for what Aurora said before about the student. You really need to believe your data. I mean, I was for many times thinking this was an artifact, and I repeated and repeated and repeated and did many, many times until I said it cannot be an artifact. It must be real because it always happens, no? So what we saw is that when we expressed the mutation of twinkle, there were some of the cells, they have these weird structures, these like reorganized membrane when something was intercalating inside the mitochondria and then the nucleoid was there. But what was that? No? So we did electron tomography. So we selected some of these regions. For example, you can see here one of these reorganized membranes, also some stretched vesicle popping out here with something inside, or here even like something that was going on from the mitochondria and something, another organelle here close by. We took a tomogram, we reconstructed, and we got this. So tomography is a little bit tricky technique. You really need to follow stack by stack all the membranes. So there were some membranes that we could not attribute to mitochondria. There was something different. There's not mitochondria, it was another organelle who was there, really close by to mitochondria and really close to the nucleoid. What was that? So then we use the other property of APEX2. We use biotinylation, purification, and mass spectrometry. And here you can see a summary. This is a very long story, make it very short. But what you can see is that in twinkle, mutated twinkle containing or affecting the mitochondrial DNA, we have a huge enrichment in vesicle proteins, all proteins related in vesicles. And among them, we have also endosomes and we have also lysosomes. Surprisingly, we don't have autophagosomes. This is something I discussed many times with Luis-Carlos and also with Julian, but we don't have autophagosomes. We have actually lysosomes there. But what is going on there? How can we just know which type of proteins are involved there? So this is a new story that was published also some months ago. So among all these proteins that we found in this our first analysis, we found two guys. We found some RAP5, and RAP5 is a protein from the endosomes, and we found SAM50, and SAM50 is a protein from the membranes. So what we thought just to know exactly what was going on here and which proteins were involved in this removal or of mitochondrial DNA, we did another experiment, which was an adaptation of the turbo ID and also proximity proteomics. So turbo ID, similar to the APEX2, is a protein that induces biotinylation of the surrounding proteins, and then you can just isolate them. But we changed it a little bit. We didn't do turbo ID, we did split turbo ID. So we split turbo ID into halves. We have two inactive forms that they don't do anything. When they come in close contact, which is, if I'm not mistaken, it's around 10 nanometers or something like this, then the enzymatic activity is reconstituted, and then the protein works again, and it can biotinylate. So what we did is just to fuse one piece of turbo ID to RAP5 and another piece to SAM50, and then try again to get the proximity proteome of this area, this contact size, no? And here you can see what we saw, no? So the first thing that actually puzzled me a lot is that we got mitochondrial matrix. We got nucleoid, mitochondrial inner membrane. How is this possible? I mean, we're talking about outside mitochondria. There are really close compartments. We have an endosome, and we have a mitochondrial outer membrane protein. However, the proteins that we are getting are inside the mitochondria. So actually this means, in my opinion, this means that there are some type of rupture of the membrane that is allowing the biotin to go inside and then biotinylate this protein. So we go a little bit later to that. Let's just focus in one guy here that we got. We got this guy here, is BPS29. BPS29 is a member of a big complex which is called the Retromer. Retromer, it's a complex which is formed by three main proteins, BPS 26, 35, and 29. All of them, they are together. They are always together. They don't care about mitochondria. You can see here BPS 29 GFP goes with the 35, goes also with the 26, and they partially co-localize with the lysosomes LAM1, also with the endosomes here in RAP5, and they don't care so much about mitochondria. So they are two different organelles. However, in some situations, and mostly in situations of stress, BPS35 is known to also work on mitochondria. What is exactly BPS35 or Retromer? I'm always maybe shifting to BPS35 or Retromer. BPS35 is the main component, no? So it's the core component of Retromer. So we talk about Retromer, but I might say BPS35. So in situation of stress, it's known that BPS35 induces the vesiculation of mitochondria, induces the formation of vesicles from mitochondria containing oxidized cargo, and this cargo might be delivered to peroxisomes or to lysosomes or to different compartments. It's what is called the mitochondria-derived vesicles, no? So they are just a piece of vesicle that pops out from the mitochondria and works in the quality control, signalization, etc. So we thought, okay, is it possible that maybe the release of the mitochondrial DNA, or what is happening in the removal of mitochondrial DNA, is going through vesicles? So that's the first thing we thought. So since mitochondrial DNA is in the matrix, what we did instead, first, what we check is the formation of mitochondrial-derived vesicles from the matrix. And how we did that is just by double staining a mitochondrial outer membrane, which is TOM20, together with a mitochondrial matrix protein, which is PDH, or pyruvate dehydrogenase. In normal conditions, we basically don't see anything. I mean, PDH is inside TOM20, and the Retromer is just over there around. So I forgot to say here, we use also in these experiments a mutation of BPS35, which, by the way, is related to Parkinson's disease, which might work as a loss of function mutation, okay? It might have other functions, but in this case, it might work as a loss of function mutation. So, however, when we stress the cells by expressing this mutation of Twinkle, we actually saw the formation of vesicles. You can see here these little tiny vesicles, which are PDH positive, TOM20 negative. We saw them in a steady state, but what was very interesting is that they accumulate strongly when we block the lysosome with chloroquine. So this means actually that these vesicles are formed. These vesicles are very fastly removed, but they are removed by lysosomes. When the lysosomes work, they accumulate even much more. So that's why you are going to be always seeing chloroquine, because it made our lab easier, no, to use chloroquine in all of the experiments to be able to see them much faster. To prove also that this mitochondrial DNA was also cytoplasmic upon this stress and stuff, what we did is just obtain mitochondria-free cells. So we just eliminate mitochondria-free cells, and we did qPCR of mitochondrial genes. Then actually we found that upon this type of stress, the number of mitochondrial DNA was increasing the cytoplasm, or let's call it non in the mitochondria, no outside the mitochondria, mitochondria-free fractions. But is this the case? So is it again going in this pathway? So what I'm showing you here, yeah. So this is an immunofluorescence about the mitochondrial DNA. So here you actually can see that in cells expressed in normal BPS35 and stress with chicken, we have a lot of mitochondrial DNA or double-strand DNA in the cytoplasm. We checked before by qPCR that we believe that this is mitochondrial DNA. This actually not happen in the mutation of BPS35. So we actually do have a release of mitochondrial DNA, which is non-mitochondrial, and it's free in the cytoplasm. But is it really free? Is the vesicle is inside? Where the hell is this mitochondrial DNA? So this is a question that puzzles me a lot. So what we decided to do, and this was done in Cologne with amazing microscopies in CCAT, was we did CLEM, correlative light and electron microscope. So how does it work? So basically we take a cell, we stain it with here mitochondrial DNA with cyber gold, we stain mitochondria with this dye here. We take a picture and then we fix it and then go automatically to the electron microscope. Electron microscope, we can see everything in the electron microscope, but we have a vesicle, we will see it. If we have whatever, we will see everything, right? That's what we did here. So you can see so beautiful here how the mitochondria, here elongated mitochondria, the cristae, and in between this cristae, we have the mitochondrial DNA lying on there. What about the cytosolic mitochondrial DNA? Is in a vesicle? It's where it is. So we kind of modify this protocol a little bit. We have cells here which express BPS35, which also express the twinkle cherry. And then we found some spots where mitochondrial DNA was actually popping out of the mitochondria. No? So it was the kind of together, not inside, but together. And we went to the EM. Surprisingly, we didn't see a vesicle. I mean, we didn't see just a random vesicle with one membrane or with no membrane. We saw something that reminds us, and actually, I mean, it is, if you are a little bit familiar with electron microscope, it's a recycling organelle. It's a lysosome. It's something that contains many recycling things, no? So you got this here. So we did also a tomogram, and this is basically what we see. So here we have here the mitochondria was popping out the mitochondrial DNA, and this very close by together, we have this particle, which is actually a lysosome. We confirmed it with the lysosome by other immunofluorescence and stuff. This I'm not showing you here. You have just to believe me, no, that this is just a recycling organelle or a lysosome. Is it an MDV or is not an MDV? We come all the times coming to the same. We struggle a lot with this question, and mostly to try to publish this paper. We finally tried to do an experiment that, okay, if this is an MDV, we should be able to see vesicles containing PDH, so the matrix, together with the DNA. And here we use a plasmid containing a sequence being linking to mitochondrial DNA, which glows in green here, and then we try to find PDH positive, DNA positive vesicles. However, we did not. We did not find this. So we saw MDVs, PDH positives. We saw DNA particles, but they were never together. They were never together. So in this type of stress that we are doing, that interfering with mitochondrial DNA replication, there are MDVs, there is a DNA, but they are not together. Actually, one of the things that we did was to interrupt the MDV formation with using MIRO1 knockout cells. And here in MIRO1 knockout cells, we still see cytoplasmic DNA or mitochondria-free DNA. So this actually was the proof that the DNA, at least the DNA, was not going out through MDVs, or at least always I say in our type of stress. It might be in other situations that it is, but in our type of stress, it's not. How is it then happening? So Luis-Carlos already talked about pores, no? And this is what we thought the next, no? So we have two types of pores which have been related to mitochondrial DNA release. And one is back and backs, which opens a big pore related to apoptosis and then release big nucleoids and many other particles. Another one is beta-guan, which they are small pores and release mostly fragments of mitochondrial DNA. So in this work, what we did is just, we didn't arrive to do it genetically, but we use specific inhibitors of the pores. So you can see here in DMSO treated cells where we have a lot of mitochondrial DNA outside the mitochondrial compartment. We use VV12, is a specific inhibitor of beta-guan, and we see still a lot of DNA outside the mitochondria. And we use V5, which is a specific inhibitor of BAX. And in that case, the number of cytosolic DNA was drastically reduced. So suggesting that actually the release in that case might happen through backpacks, macropores. Just once I come back now, this is actually something we just published now, but already in 2022, we already saw something similar. So here we were using ethidium bromide to damage the mitochondrial DNA and to release the mitochondrial DNA, as it has been shown before. And here in super resolution images, we can see that in these conditions, we could see BPS35 together with SAM50, which is the membrane, and together with BAC, all together in the same spot. So again, for this type of stress, which is mitochondrial DNA replication stress, we end up in a model where we actually have a parallel pathways. In one way, we have the activation of canonical MDBs, which contain mitochondrial matrix material. They go to the lysosomes, and they might be signalizing that the stress is happening. At the same time, the lysosomes, they are coming closer to the mitochondrial surface, and there is a pore which is opening, and this DNA is automatically taken up by a lysosome. It's not free in the lysosome. Mitochondrial DNA goes into the lysosomes. We believe, of course, for degradation. But we didn't stop there, no? So it's like, okay, these are very beautiful, but can we use it? Is it really useful? So remember what I showed you before. So in 2005, already it was said that mitophagy might control the accumulation of mitochondrial DNA. This has been used in the field, so you might recall rapamycin, for example. You might recall also metformin. So this drug that we are using right now in to treat many diseases, they have been seen to also activate mitophagy. However, the role in mitophagy, even though it might look beneficial, for example, there is a clinical trial against aging using rapamycin. Also, the long-term use of these drugs might cause some problems because you are also activating many things. But what about this pathway I just described? It very looks specific to the mitochondrial DNA. Is it possible that we pushed up this pathway to clean up mitochondrial DNA? So, of course, it was very preliminary, but in that case, we used Drosophila. We generated Drosophilas, larvae Drosophila with a big deletion in the mitochondrial DNA just by expressing a restriction enzyme together with a DNA ligase in the mitochondria. We cut this piece of gene containing cytochrome B, and then we got two types of molecules, one wild-type molecule and one molecule containing a big deletion. So this is just the confirmation we have the deletion here. How do these larvae look? So you can see here, these larvae, they look bad. So I'm not a Drosophilus. This was a collaboration that we did. But this, I was told that this was stress. I mean, these larvae, they were so stressed. They reached the pupae stage when we grow them at really low temperature, but they didn't hatch. So there was no adult flies here. At the molecular level, what is interesting for me, we actually have a reduction in mitochondrial DNA copy number, and we also have a reduction in the ratio between these two genes, indicating that we actually have heteroplasmy in this gene. So very easy in Drosophila, as you might know, it's very easy to just upregulate and regulate genes. So we just upregulated BPS35, so the component of the retromer. You can see here, we make it up on average around 5 times more of the wild type. Also here, the confirmation that we still have the deletion in mitochondrial DNA and everything. What happened then? So larvae, they look cool again. So the signs of stress are gone. So we actually could, at least macroscopically, remove the damage that was caused by mitochondrial DNA. Let's go deeper, also into the electron microscope. You can see here that the damage in the mitochondrial DNA causes these big changes in the mitochondria with completely aberrant crystal, accumulation of autophagy or let's say lysosomes in that case. And when we express BPS35, mitochondria goes back to normality, or at least what we believe is normality. The molecular level, again, we recover mitochondrial DNA copy number, we recover the ratio, cytochrome B, ND5, so it's actually beneficial. And not only to the mitochondrial DNA, there are many other pathways that might be activated upon mitochondrial DNA damage. So we did a transcriptome analysis of this larvae. There's these 5 or 4 pathways which are activated, including innate immune response, damage to oxidative stress, glutathione metabolism, and transmembrane transport. So we selected some of these targets which were actually regulated when in larvae containing this mutation, and all of them come to normality upon expression BPS35. Okay, this is still a very early experiment, but I think it's actually a proof of concept that we can actually modulate mitochondrial quality control to improve mitochondrial health. And just going back to mitochondrial quality control, this is a very recent scheme of the number of the different mitochondrial quality control pathways that exist. So it's not only canonical mitophagy, there are many other pathways that you can see here, MDBs, MDCs, spots, mitochondrial transfer, extrusion, and I believe that we will find many more. For the mitochondrial DNA, it was described in the 22 in the germ layer in Drosophila. It was described again, so more or less the same time, 21 also in yeast by this Christoph Osman, the paper we show about the sphere of influence. We also show that it also happens in mammals by direct transfer to endosomes and then lysosomes. It was also confirmed afterwards by another group. And also now really recently, there was another pathway specifically for the inner membrane of the mitochondria. So this is just the take-home message I want just to tell you. There are many quality control pathways, still many more to discover. And the good thing, on what at least interests me, is that we can use them. We can use them actually to do something good. Of course, we are very preliminary research that might be in the future some trials to prove that. And with that, I just want to thank you. And now just to say thanks to the people who give me the money to do this research. This work has been done in Cologne, and even though I'm in Düsseldorf. So it's really worth to say that mainly this work was done in Cologne and continue here in Düsseldorf, where I am located right now. This is just 35 kilometers away, so I didn't go so far away. I'm still in Cotton with all the people of Cologne, and that's it. And thank you very much for your attention.
Thank you very much, David. Wonderful talk. So, any question in the audience? Let's give some time. I have some questions, but
yeah.
I will. So, both of you are using mitochondrial DNA replications errors.
Mm-hmm.
So what happens with point mutations? What happens in a tRNA mutation? What happens in a complex one mutation?
Well, that's a very good question. To be honest, I don't know. I mean, I don't know because I never tried it. The stress we are doing is a very specific stress, no? And for example, when interfering with the mitochondrial DNA replication machinery by depleting TFAM also leads to the release of the mitochondrial DNA. For me, it looks like it's a quality control during the replication machinery, no? So kind of mitochondrial DNA replicates, and then something is not going okay, and then there is a pore opening and releasing whatever is bad. How a specific mutation in the mitochondrial DNA affects this pathway, to be honest, I don't know. I mean, I don't know. I would.
So my next question will be, have you tested POLQ mutants or OPA1 mutants that they show mitochondrial DNA depletion? Like,
so we are, yeah. No, we have not. We have not tested that. So we are working mostly in the mitochondrial dynamics proteins. Have to check that. But no, we have not. But I think it's a good idea. Maybe we should go again to.
I mean, because of this POLQ paper from Anu that show there is this release, but no one have shown how it is. And since there are a lot of mice models now, have been just last week published.
Well, I can imagine that a Pol gamma the mutator mouse or the Pol gamma mutation, it might do something similar. But this is something I'm just speculating. I don't have the proof for that.
So, any other questions? I could be asking questions all the time, to be honest. And what is next?
What is next?
Yes.
Well, I think we are going to be, our group is going to be a very nice competitor with Luis-Carlos because we work in similar things. I mean, I'm very interested as well to understand how mitochondrial DNA is recognized, no, when mutation. But at the moment, we're mostly focusing in how the cell reacts to mitochondrial DNA stress. So we are trying to do other type of experiments to focus more in cell effects more than a mitochondrial effects, no. But anyway, what is next is just to understand. Of course, if we can use this mechanism in mammals, for example, if and how the cell reacts to the stress. So not only by
Yeah.
removing this DNA, but I don't know, activating other pathways, no, that might be
So
complementary to this.
we have a lot of data in different mutants with different stress, and each mutant reacts totally different, and even the same mutant in different amounts of heteroplasmy or with different nuclear backgrounds, so from different patients reacts totally differently. So the complexity is just escalating very much, to be honest. That's how I see it. So it's just this, for these mitochondrial DNA patients, trying to do personalized medicine will be always the key, because otherwise it's very difficult. And also, since each tissue will have a different amount of heteroplasmy, etc., there's little observations that might be, that's why I was very intrigued by the fact that if you were testing these twinkle mice that were tissue specific with the same insult, whether you get the same result in different tissues. Do you?
Well, we have our lab. So the mice are just tissue, but in the cells we don't combine what we are doing in the cells with the mice. Eventually, if we do some type of primary culture, we might also try there. Anyhow, I think both pathways, what Luis-Carlos showed before and what I'm showing you, they are complementary. So I believe, for example, if we come back to this theory of the sphere of influence, is it possible also that one mitochondrial DNA containing a mutation is affecting one area of the mitochondria? This area is kind of divided by fission, no, and isolated in a specific area of the mitochondria, and then eliminated by a normal autophagy so they are just complementary pathways. I think the difficulty here is just to see it live.
I can
can
jump into it. I totally agree with David. So I think that the different pathways can be activated, for example, depending on the amount or the proportion of molecules that are damaged. Imagine that when you have a small damage, let's say less than 5% is the lysosomal pathway going on, but imagine that you surpass certain threshold, that you have a big damage in the mitochondria in a big area. So in that situation, maybe it's mitochondria or mitophagy to get rid of more molecules. So maybe it's like, I think all this, because it's not only about this pathway or the pathway we describe, it's also the BDI in papers about lysosomes. There's many other pathways. So of course, I think in science we should not have a very focused idea. It's like multiple things can be happening at the same time, because normally we used to focus on my model would explain everything, and it's not like that. Even me, like I'm sure that I've seen that even in our model there are things going out through independently of autophagy, so it's perfectly
nice. Okay, so if there is no other questions, I will have to say thank you very much. It's been a pleasure to have you. We hope we will see you both. I think Luisca is coming to the, is another one, to the meeting of the SEBBM this year in Cáceres. We will have a very metabolic session in the mitochondrial with Laura Formentini and Lorea Barcárcel. Okay, and thank you very much. It's been a pleasure.
Both of them.
Thank you.
Thank you.
Thank you. Thank you very much.
Thank you. Bye.
Bye.
Ya estáis fuera.
Vale.
Que-
Estamos fuera ya.